I will maybe speed up our presentation. Okay. Good afternoon, everyone. My name is Zheng Chen. I'm an assistant professor in Department of Computer Science at Osaka University. Today, together with my colleagues, we will introduce our recent work on an AI agent system for automating the gene annotation function task. And I will give some introduction and background of the AI agent, and we will present how our tool works and share our findings. Okay, this is today's presentation. We will first briefly introduce our team and our motivation, and we will give an overview of agent-based AI system for the bioresearch. Then we will present our tool, including the pipeline and how to use our online tools. After that, we conclude with a case study demonstrating its application to the microbiome strain analysis. Okay, this is our first time to attending this conference, and we are startup research team in academia, and our members include some researchers and PhD candidates from computer science background. We also have Zui, and he is a incoming assistant professor at the Bioinformatics Center of Kyoto University. Actually, where the KIC team is also based on this center. We collaborate with a researcher from the Bioscience department at the University of Tokyo to support our AI model evaluation. Our team focuses on the building AI infrastructure and AI models for bioscience, biodata science. Actually, today, as AI is booming, every week there are several new tools are released and proposed. However, evaluating these models remain a challenge. Like the last speaker, he mentioned maybe as an AI researcher, we are hungry for data. However, it's very challenging for us, mainly because the AI models typically require suitable training and testing data site, where there are several open portals for the biological data. Curating data resource from such biodatabases is still difficult for us because this step requires domain expertise and experiences. So our team tried to create machine learning ready data sites to provide the pretty precise formats for the similar training and evaluation. We also benchmark some AI models on the oncology multi-omics data sites such as TCGA. Maybe you can check our paper. It's recently we published a paper in the Nature Scientific Data. We benchmark whole the TCGA database to the to the machine learning researcher. They can directly call our data site and develop and evaluate their AI models. Also, the bio researcher maybe they can easily to explore the model capability and choose the best one based on their preference because we benchmark several the high cited published models. Then we are also developing some AI tools to automate some time-consuming tasks such as data processing or the integration. We also focus on the formulating some omics data analysis research as solve machine learning tasks and deliver some AI-driven solutions. And we also target some top AI conference like this ICLR or NeuroNIPS. Okay. Okay, today I will introduce 1 hour written work, and this work is based on a new paradigm of AI agents. And we will present how we apply it to gene function annotation. Actually, the machine learning models are task specific and often rely on the structured data and manual feature engineer to extract some statistical features. For example, sometimes if we want to analyze the data resource from the multi-omics layers, we need to carefully design a data processing and integration pipeline. Sometimes this model are commonly used, but their feature representation ability and flexibility are limited. Then come to the deep learning model, which brings a breakthrough in handling unstructured data, because we can develop some encoder and handle some specific modality, and we can directly input the different omics data for integrative analysis and extract more informative features using the maybe neural network and some new training loss, powerful training loss function. But deep learning model is still the task specific. For example, giving some task, we need to carefully design the architecture of neural networks and set up some loss function to formulate this problem. And also we require a high quality data set to train a good model for the generalized performance. In contrast, there is a new paradigm, the agentic AI. The behind of this paradigm is the foundation model. Foundation model means we already train a model using the DDS and diverse database, and this model already provided by maybe the top AI companies, OpenAI or Anthropic, and we can directly use the model and fine-tune this model for some specific task. So this model are not just to train to solve a specific task, and so this model this paradigm is tax-free the AI models and acts as a script to automate a workflow by using diverse external the tools API or the knowledge database. For example, today I think everyone used the GPT. When we use the GPT, you may find you can correct their behavior based on sometimes you directly interact the interface and give the GPT your feedback. So the AI agent system also take advantage of this behavior, and so they have the memory if you directly to give some good reward function or give directly some feedback, so they can perform the task based on the human preference. So as shown on the right, the figure, this system interact with three key components: the users, the environment, and the external tools. They reason, plan, and act. Often through the API or UI interface based on structuring and instruction and behaviors. This make it as a AI agent is a promising direction for automating the workflow and the pipelines. Okay, here I give an example. How does an AI agent help with biological data analysis? So one key capability is to go decomposition. The agent can take a high-level instruction, like, okay, analyze this data set and generate a report. Then it can automatically break it down into logic subtypes, like the loading the data and the processing, analyze, or visualization. Also, we can adjust these subtasks based on our preference using the language model. Once the tasks are decomposed, the agent follows a circle of reasoning and execution. It starts with the thought, like asking, "Oh, what should I do next?" Or then it's take action, executing selective steps, and finally perform observation, evaluating the outcomes. This interactive loops allows agent to dynamically adjust its plan and do a reasonable execution. Okay, then maybe we will give an introduction on how do we use the agent for bio case study.
Okay, so next I will introduce our cases about how we can use the AI agent to help us to process our daily task. So in these studies, we will work with newly sequenced microbial strains. And a key challenge is understand what their genes actually do, especially since many of these genes, they don't have any functional annotations available, right? So functional annotations using a database like the KIG helps to link the genes to the metabolic pathways and is key for the downstream analysis. And our goal is to annotate these new genomes by comparing them with the well-studied reference and the reference strains that do have the KIG data. To do this, we first use the KIG to identify a suitable strain as a reference and use them genomes to which also has the annotated pathway data. And then we retrieve the genome of the reference, including the gene antenna first and the other necessary informations like the nucleotide sequences, maybe from the open source database like the GenBank. And finally, we can use the computational tool like BLAST to align the genes from our new strains to the reference. And this allowing us to transfer the annotations where there is a strong sequence identified. And this workflow shows that this allows us to functional annotate the uncharacterized genes and even in the novel or underexplored sequencing strains. And our motivations that we has introduced comes from a common need. We compare the gene context across many strains. And we often need to align and analyze thousands of genes across multiple genomes and trying to understand the functional differences and what every gene does. But doing this manually is increasingly repetitive and time-consuming. Well, public database like the KIG or GenBanks, they provide the necessary information, but access it at scale is difficult. And here are some issues when we try to do this. The first thing is most database public APIs, it imposes a strict rate limits, like only it allows you to use 3 or 3 to 10 requests per second, and also you risk getting your IP temporarily blocked, right? And secondly, even if we have downloaded the data, the gene data we need in bulk, and the raw field formats across different databases can be complex, and this also makes it hard to parse efficiently. And also, we need to screen the genomes based on the specific criteria. Like in some research, we may focus on whether it has a toxicity, or we care about their host organisms, or like here, they said that we care about whether it presents of certain pathways in some database like KIG, and it's not directly supported by the original database. And this requires the manually reviewing the metadata, and we need to check about maybe the references or writing some custom filters to link the different databases through this. And to do this, this is how the AI agent tools can help us. Our pipeline uses modular AI agent frameworks to process the gene functional annotations in the new microbial strains. And our usage can begins with a simple input. This can be a strain name, the strains you are studied, or you have a genome IDs like session IDs from the genome banks, and you can just use our tools to search for it. And from the input, our first goal is to find a high quality reference genome. And here we have the reference genome screener agent. It queries the public database to retrieve the metadata, such as the available annotations, maybe also some text minings in the related references, and the associated data entries in the database. And especially it searches for the closely related strains that already have some functional annotation, for example, from another database like KIG. And this serves as the foundations for all our downstream processing. And meanwhile, we have the code generated agent. It builds a custom script that parses the fields we download from the database and extracting the necessary informations, like we may care about the locus tag and maybe the nucleic sequence data from the choosing genome. And also, And we have a data fetching and the extractions agent. And this agent addresses the performance bottlenecks in retrieval the large-scale data from the open source database. And we can use it to accelerate the process. It handles the database accessing the rate limits and manage the parallel download and ensure the stable fetchings, even for hundreds of the genomes. And after the data collections, we have the data formatting and the export agent to standardize the output. It cleans up the messes and maybe some inconsistent database fields, and it can transform the data into a structured format and tolerate to the different users' needs. And the final output is ready for the downstream analyze, such as we can directly run in the computational comparison, the BLAST, and maybe just mapping the genes to the known database metabolic pathways. So this is the AI agent tools, and altogether these agent-based tools enables the end-to-end gene annotations and the comparative analysis of the different microbial genomes. It transforms what was once a tedious and the manual process into a fast, automated, and scannable pipeline. So here I can show some details of our tools interface, the current versions and the used cases. As we said, the input and the queries can simply start with you input some of the organism names, or you already have some database you want to use, and you have the session IDs, and you can directly search for it, and our agent will search and filter the data, maybe based on your settings, like if you want to link to another database like Cake, and they just give you the results that only have the related interest in the database, and it show you the search results, and you can give a glimpse at what you already have through the first step. And next, you can give a processing result and to download the data you need. Like you can choose and customize what's the interest, and also the some dimensions should exist, or you don't need. You can just customize them and choose to download the data, and this can be used in the next step. Like you directly use the data you gain from the our agent, and you can upload your fields and to run the annotations analyze. And this is based on the customized computational tools, or you can use the simplified tools like the BLAST, and you can use the original settings like you already do. And here is the examples of the result report. As we can see, the input can be simply on chartless locus tag, which is from your nuke sequencing the genomes. And we can see that you have nothing to know about it, and whether the gene is functional, we don't know about it. And for the output, it already mapped to match the functional annotated reference genome with the match scores and the multiple result options available. Like here's some research in our teams focus on whether the associated enzyme functions exist. So we add some course to another database, like we show the EC numbers or something. And also we can group the result by the matching scores, and you can like set in your threshold and filter it. And the additional tools can be directly used in our platform for the pairized comprehensions for the strains genomes. You can identify the shared or the unique genes, or highlight the difference in the functional pathways. So here is what you can get from our tools. And thank you for the attention. You can access the tools about these links and contact us through the emails. And like we said, our team also works on the machine learning ready database and process the pipelines for the biomedical data. And also we work on the AI modules to automate the data analyze task and the data-driven solutions. And we welcome the collaborations, and thank you.
Thank you so much. So questions. Before I go there, is Prashant in the audience? Is the next speaker in the audience? All right. I don't see a hand up for that. So here we go with questions.
Thanks for the talk. I thought it's a particular example that you gave. You seem to have quite a specific,
Yeah,
fairly standardized pipeline that you were looking at.
yeah.
So I was wondering how much the agents actually give you in this context, because you kind of know what you're going to be doing already.
Yes, I see. Thank you for the question. So, yes, in these examples, we just show a demo that is you can say a highly customized task, right? So actually, you can keep the core steps like you always need to access to the database, right? So this agent is a modulate, and these modules can be kept, and you can change, like you can directly ask the agent to, if you can do something for me, and it can generate the code. And in some cases, if the code, if the task is not that like complex, it can finish because we have the code generator agent. And I think it can automate to search for if there are any exist toolbox or some code from the Python package, or maybe from another code environment, like we have already established, like the tools that can tolerate your needs from the MATLAB or R societies. Okay, it can just exclude the code for you, and maybe hopefully it can like achieve the result you want just through the same platform. Okay.
I had another comment actually. Thanks for that. You know, I think these methods, these agentic AI methods, are going to be really great for doing fairly well described types of analyses that occur in the literature that the AI can pick up on. And it's going to be really useful for a lot of wet lab biologists and you know, people who don't know what bioinformatics. But myself, as a bioinformatician, what I think I would really like is an agent that can install old, poorly documented software for me and know how to convert the output of one tool to the input of another tool, again, where documentation is so important. Do you think that kind of thing is feasible, and even though unglamorous, that people will work on it?
Yes, I think it is a really interesting topic, and actually we haven't have a try on it. And maybe if you can just send your needs to us, and maybe we can try to like to test whether this kind of task can be achieved in our environment. Okay, thank you.
Just a question online. How frequently does the AI agent hallucinate and propose wrong or artificial functions, and did you measure this?
Oh, sorry, can you repeat that?
Sorry, how frequently does the AI agents hallucinate and propose wrong or artificial functions, and did you measure this?
Oh, I think I am not currently. We didn't explore this, and maybe I can just review you and answer your questions maybe later. Sorry.
Yeah, this is from the other fair online.
Okay.
There's a question. Hi, thanks for the talk. In your slides, I noticed there was an option where it said include DNA sequence, which was ticked.
Yes.
What happens if you don't take that? Does it consider the proteome?
Okay. Yeah, thank you. So actually, in our platform, you have many customized operations available. Like you can choose you want to use the nucleotide sequences, or you need to use the amino acid sequences. And the thing is, yes, so you need to match based on some input data. It's not direct. It's not must be the nucleotide sequences or other kind of sequences. You can match based on the, like we have some natural language descriptions of these genomes, and you can just try to match if you have the gene symbols, and you can match based on the descriptions. But I think the always use the sequences data as the input is recommended.
And I also wondered when you said new strain, is it actually a new strain, or is it like a new sequence that the researcher is querying? Because if you know it's a new strain, that's already pretty cool.
Yes, so thank you. So yes, here the new strains actually means that if you have some, maybe you already have some sequencing data existed in the database, but it has nothing like to annotate its functions. It just like provide the basic like look tag, and you do not know whether it genes it functions as yours, like maybe there are some very static references. So I think in this case we call it a new string.